All right, so thank you so much for having me here today. Um, I'm going to be talking a little bit about some of the research I did during my PhD, which focused mainly on kelp gametophytes. And this specific bit of research that I would like to talk about today is about the grazing of gametophytes in a warming ocean. And I'll get to, what exactly all that entails later. But I thought I would maybe introduce myself a little bit first. Let's see. So about me, going from working on microalgae to the micro side of kelps. So I started off at Utrecht University in my native, the Netherlands, where I'm from, where I worked on fossilized dinoflagellates, and I used them to date fossils for my dissertation. But after a while, I sort of realized that I liked live algae more than dead algae. And so I moved on to NUI Galway in Ireland, where I worked on red microalgae, and specifically microalgae that grow on plastics. And so I did a little bit of work on their cell walls and how exactly they grow on plastics and sort of what that means in the context of plastic pollution in the ocean. And then I sort of promoted from working on unicellular things to multicellular small things. And that's how I arrived at kelp gametophytes. So I did my PhD in sunny Coffs Harbour in Australia at Southern Cross University, which I finished just a few months ago, where I worked mainly on kelp gametophytes. And right now, current location, I'm in Oban in Scotland, where I moved just about 2 weeks ago. So going from Australian summer to Scottish winter has been quite a challenge. But here I am, and Oban is very beautiful. And I started a postdoc at the Scottish Association of Marine Science, SAMS, where hopefully I'll be working on seaweed life cycles in the context of both climate change and seaweed cultivation. So, what I'm going to be talking about today is, as I said, about my research that I did with Southern Cross University during my PhD. So for people that have been to I2S, it's going to be a similar talk to what I did there. So it might be a bit repetitive, but hopefully there's lots of new stuff for new people coming along as well. So I'm going to start by talking a little bit about kelp forests and what gametophytes are and what their ecology entails. And then specifically, I'm going to focus on the grazing of gametophytes. And we'll also look at how gametophytes can even survive in poo. And then hopefully we'll have some time to think about what it all means. So starting off with kelp forests and gametophyte ecology. So kelp forests are foundation habitats. And basically that means that they provide this really wonderful 3D structure in which lots of species can live and thrive, and that way they support a lot of biodiversity. So this is what it would look like around the coast of Australia, where you have Ecology radiata being kind of the main kelp. And you can encounter anything from fishes to, wobbegongs, seals, or sea lions and lobsters, lots of different iconic species. And through that, they support things like tourism, and they're great spots for recreational fishing and diving. And so they also represent huge economic values. But sadly, kelp forests are declining, and specifically also in Australia. So this is the map of Australia, and here in green, you see sort of the distribution of Ecolonia, and you can see that it pretty much wraps around the entire southern half of the continent. But there's several reasons why it is declining. So one of them would be heat waves. So I think currently there's a massive marine heat wave coming to the east coast of Australia and Tasmania. And there's been a couple of bad ones in the past as well. And so they have really, impacted Ecolonia in certain places. You also have bad water quality, and urbanized environments can really have a bad impact on local kelp forests. And then there's obviously ocean warming, so the gradual warming of sea surface temperatures, which can cause range contractions in kelp. But it can also facilitate the moving of certain grazers into temperate habitats, where they can then destructively overgraze the kelp. And so sort of zooming in on this grazing, which can be quite a problem for kelp. So what happens when kelp gets grazed? It can sort of fall back into two alternative states, one of them being algal turf flats. So you go from this beautiful lush kelp forest, and after the disturbance, these small filamentous algae will take over, and they trap heaps of sediment, and you really lose like that beautiful 3D habitat. The other one is the urchin barrens. So urchins can overgraze kelp forests. They don't always do it, but they can do it. And when they do, you will basically be left with just bare rock with calcifying algae. And so, algal turf flats and urchin barrens are different systems, but they have some stuff in common. And one of them being is that they represent loss of biodiversity, but they also stay in place after the kelp leaves, by somehow repressing recruitment of new kelp. So once it's in place, it's quite hard to get rid of these sort of degraded environments. And so, when you think about grazing being a problem, and when you think about the fact that recruitment is repressed, you kind of end up with the gametophyte. Because recruitment of new kelp happens via the microscopic gametophyte. So kelps have this biphasic life cycle where you have two separate living stages. One of them is the sporophyte, which is the adult kelp plant that you see out in the ocean. They can become fertile and make fertile tissue that produces spores, which will disperse and settle, and they can grow into gametophytes. So they are the free-living sexual phase. So you can have male and female gametophytes that produce produce eggs and sperm. And when they fertilize each other, it will go back to a sporophyte. However, they don't have to do that straight away. So they can sometimes also grow vegetatively, and they may be able to do that for who knows how long. It's really hard to actually study them in situ because they're so small. So they sort of, refer to as this big black box. So there's lots of stuff that goes on there that we don't really know about yet. And so a while ago, me and a whole bunch of other people did this review on everything gametophytes and ocean change. And, there's a lot that goes into that review. But the thing that I wanted to talk about today is the fact that out of a database of about 200 papers, there was only 10 that dealt with the actual ecological interactions of gametophytes. So, for some species in some places, we have a pretty good sort of sense of, what temperatures they like or what other abiotic factors impacts them. But how they interact with other species is a bit more of a knowledge gap. So having that knowledge gap there and knowing how much grazing impacts adult kelp forest, I kind of set out to ask, well, is there anything grazing the gametophytes that might impact that recovery? So specifically, my question was, do any grazers occurring in either turf flats or urchin barrens contribute to that repressed recruitment that we see? So the fact that after kelp disappears, it's not really able to grow back. And if that is the case, is it exacerbated by climate change? So to test this, I used 3 different grazers from different sort of ecological niches in Australia. So we have the turf grazer, Amicus atkinsoni, which is quite cosmopolitan. So both in tropical and temperate Australia, you can find them. Then we used the juvenile of a tropical grazer called Trypnoustis gratilla, which doesn't interact much with kelp, or I should say, hasn't really been reported to overgraze kelp, but is also hypothesized to move further into kelp habitats as ocean warming sort of continues. And we used the juveniles of the temperate grazer, Centrostephanus rogerisi, which is the urchin species that in Australia is mainly responsible for overgrazing kelp down south in Tasmania. So I settled a bunch of gametophytes to test this, and then I added either grazer or no grazer as control. And then I used 3 different temperatures to test the influence of average winter, average summer, and future climate change scenarios, equivalent to Coffs Harbour, which is where I sourced the gametophytes from. And then I used 6 different time points because I wanted to be able to see if the gametophytes could recover after grazing. And then that was replicated 5 times. So first we'll look at the direct impact of grazing. So basically what we saw is that every grazer that I used lowered the abundance of the gametophytes after only 1 day of grazing. So here you see on the y-axis the abundance, and the blue bars is the control, and red is grazed. And you see that both the micro snail, so the turf grazer, the tropical urchin, and the temperate urchin all reduce the amount of gametophytes significantly. Interesting enough, though, no apparent effect of temperature on grazing. So regardless if it was 19 or 26 degrees, all three different species of grazers ate about the same amount, which is quite interesting because they have quite different ecological niches. So we kind of would expect an effect of temperature, but that didn't happen. However, how the gametophytes were able to recover was impacted by temperature. So here I'm going to mainly focus on the two urchins. So for the tropical urchin, Trypnosis gratilla, we're looking at 32 days post-grazing. And here on the y-axis, again, you see abundance. And at winter temperatures, 19 degrees, you actually see recovery. So an increase in abundance in the grazed treatments. At 23 degrees summer, you see no recovery. The grazed red bar is significantly lower. And then you see an overall decline of gametophyte cultures at 26 degrees, which is the ocean warming scenario. So you see here at 19 degrees, abundance actually goes up, which is a bit weird because, we obviously didn't add any extra gametophyte to our cultures. But what also happened at the same time, which is not pictured here, is that the length of the gametophytes decreased. And so our sort of hypothesis was, well, maybe the way that these tropical Trypnosis eats the gametophytes is by shredding them, and then the little bits and pieces that stay over can regenerate and grow into new gametophytes at 19 degrees. We saw kind of an opposite effect with the temperate grazers Centrocephalus regerisi in the recovery phase. So here again, we're at 32 days post-grazing, abundance on the y-axis, and you see here that there was no recovery at average winter temperature, so abundance has stayed low after grazing. There was an overall decline of cultures at summer temperatures and also an overall decline at ocean warming temperatures. So again, here, abundance stayed low at 19 degrees, but again, in the background, the length actually increased. So what we think might happen here is that maybe Centrocephalus eats sort of the gametophyte whole, and then that sort of reduces the density of the cultures, which might facilitate the survivors to grow bigger. But, it's a bit of guesswork here, but it's interesting to see that the different grazers had different effects on the recovery phase of the gametophytes. So that was sort of what happened when we let these little critters graze the gametophytes. But what was really interesting is that we actually saw the gametophytes surviving inside the poo of the grazers. And it might seem a bit weird, like why would I even measure that? But when we first saw it, it was sort of anecdotally. So, I had all these grazers and I was making them eat gametophytes, but I wanted to make sure that they actually did eat them. So I checked their poo for gametophytes. And I actually found them back in there, alive and well and thriving even. So some of these gametophytes were actually recruiting in the poo, making baby seaweeds. And so, like I said, we first saw that with the turf grazing experiment, which was the first experiment that I did. And here you see juvenile sporophytes growing in the in the snail poo, healthy gametophytes and spores as well. So every single phase of the gametophyte life cycle sort of was able to survive being eaten by this micro snail. And that was obviously very interesting because that's not something we expected. And it was quite cool. So there must be something beneficial to being ingested for these gametophytes to actually make babies, so to say. So when I did the series of urchins experiments, I sort of set out to test this a bit more, rigorously. And here we saw the same effect again. So survival and recruitment in poo of the tropical grazer Trypnosis gratilla. So here on the y-axis, you see the number of sporophytes growing, and the blue bar is the ingested treatment, so the gametophytes in poo. And you see that there was a really high recruitment in poo at 19 degrees. Also interesting to see is that there is no recruitment in the control treatment. So control is yellow bars and there are no yellow bars. So the only sporophytes that, appeared in these cultures were the ones inside the urchin poo. So there's an urchin poo and there are some sporophytes. But this didn't happen when we tried the same thing with Centrostephanus regersii. So ingestion did not seem to facilitate recruitment. So we saw zero sporophytes in any of the treatment. But that was despite plenty healthy gametophytes. So they did actually survive the ingestion. So here you see the proportion of dead and alive gametophytes after being ingested by Centrostephanus, and you see that 50% survived in every temperature treatment. So there's something going on there that stops them from recruiting inside the poo. So what? Why would I care about, tiny seaweeds growing in tiny urchin poo? Like we're talking, really, really small. So kind of going back to, the original research question, do any of these critters help repress recruitment of kelps in degraded environments? And well, the answer is, it kind of depends the habitat. Different habitat, different response. So we saw that turf grazers could actually facilitate recruitment, which is quite interesting because, if you go out there and you take some turf algae out of the ocean, there's actually a lot of stuff growing and living in it, very small little species. And we only isolated one of them, but we found that they could actually help gametophytes fertilize. And we saw the same effect with Trypnoustis. And when you think about it, if you're a gametophyte out there in the ocean, you need to be at least 1 millimeter together in order to effectively fertilize. And being eaten by all these small critters and getting all mixed up in the gut, you can imagine that actually helps like finding a mate. And so when you think about it, there might be a real role there for small critters, which is quite interesting. Especially in the case of Trypnoustis, because, as I said, they're not really known to overgraze kelp forest, but they have been hypothesized to move further into kelp forest as ocean change sort of continues. But here we see that there might also be some kind of positive interaction. So yes, it would be quite interesting to sort of investigate that further. However, Centrostephanus regersii, we didn't see that recruitment effect. We just saw that they could eat the gametophytes and then nothing happened afterwards. And that's interesting because Centrostephanus, as I also said, is actually the urchin that is responsible for overgrazing down in Tasmania. So, it's only one study, so it's not, it's a bit too early to put any big conclusions on that, but it's definitely something that warrants further investigation. Should also point out, though, that at every level of ocean warming, every level of grazing, there was never any recruitment at the ocean warming temperatures. So, no matter how beneficial anything may or may not be, when it gets too hot, gametophytes stop recruiting. However, I still think there is, sort of a positive message out of this, and that is that of the resilient gametophytes, because we saw that they can regrow after grazing. They can survive and even recruit after being ingested. And even at the highest temperature treatment, there was always some that did survive. So they may not be recruiting, but they're, hanging in there, which means that, in this sort of vegetative state, they might be able to survive some really high temperatures and, be ready to recruit when temperatures drop. So that's all that I was going to talk about today. So thank you for being here, for listening.